Recording in progress.
अस्सलाम वालेकुम वरहमतुल्लाहि वबरकातुहू। अस्सलाम वालेकुम।
Walaikum assalam.
How are you?
I'm good. How are you?
Alhamdulillah. Is my voice clear?
Yeah.
Okay. So today we are going to start with chapter number 4 of the biology, that is cell membrane and the transport. And this, if you see the content, in the content they have mentioned that the fluid mosaic model introduced in 1972, which we will discuss today, describe the way in which biological molecules are arranged. Now, in this chapter, we will discuss, according to the fluid mosaic model, how different biological molecules like glycoproteins, glycolipids, cholesterol, and integral proteins, how they are arranged in the cell membrane. And then we will discuss the model continue to be modified. We will discuss in detail what are the modifications, what is cell signaling, all these things. And 4.1 section 4.1 deals with, like, you must know after completing this, that fluid mosaic model structure, the hydrophobic and hydrophilic interaction, and phospholipid bilayer formation, then the arrangement of cholesterol, glycolipid, glycoprotein in cell membrane, like how they are arranged, how cholesterol is arranged, how glycolipid is arranged, and whether they move inside that membrane or whether they are stick to their position. So this we will discuss. And in the section 4.1 part 3, there is discussion about the role of these. Firstly, in part 2, we will discuss the arrangement of these things, the arrangement of cholesterol, glycolipid, glycoprotein. But in part 3, we will discuss their roles as well. That what is the role of phospholipid, what is the role of glycoprotein in the layer, in the phospholipid bilayer? What is the role of integral protein? How they help in transportation? Okay. And then in section number 4, section 4.1, part 4, we will discuss the transportation of these things. Like how different molecules transport by the help of these molecules, by the help of glycolipids, by the help of glycoproteins, how different molecules enter the cell or how different molecules leave the cell. Okay.
Okay.
Okay. So in the section 1, that is introduction of the cell membrane. In this section, if you see, firstly there is the discussion about the phospholipid. Whenever we talk about the cell membrane, there is a discussion about phospholipid, and we have discussed it in detail in the chapter number 2 in the biological molecule. That phospholipids are, they are lipids, but there is one difference from the other lipids. If you see, this is a phospholipid, and we have discussed it that in phospholipid you have a fatty acid tail. This hydrophobic tail is fatty in nature. This is fatty acid tail,
Yeah.
and the this one is hydrophilic head. In the hydrophilic head, you have phosphate group present. right? This we have discussed
Yeah.
in detail. So this is the basic structure of the phospholipid. And when we see how phospholipids are formed, firstly, we have discussed this in the biomolecules in the section of lipids. That firstly, in lipids, the most important type of lipid is triglycerides, if you remember, the triglycerides. And how
Yeah.
triglycerides form? By one molecule of glycerol and three molecules of three longer chains of fatty acid, three hydrophobic chains of fatty acid. When they combine, they form. यार मेरे को wait a minute, Michelle.
Okay.
Okay. So I was telling you that there is one glycerol molecule and three fatty acid chains, and these are hydrophobic in nature. And we have discussed in detail. There are two kinds of fatty acid chains. What are those? The saturated fatty
Saturated,
acids.
saturated and unsaturated.
Yes, saturated fatty acid chain, in which there will be. What will happen? What is the composition of saturated fatty acid chain?
Saturated fatty acid chains,
Hmm.
um, they have, they don't have any double bond, carbon bond, double bond.
Yeah.
And
All the single
unsaturated,
bonds are there.
yeah,
In the carbon-carbon, in between carbon-carbon, there are all single bonds. And in unsaturated fatty acid, what is the
they
composition?
have they have carbon double bond.
Sorry. And in
Yeah,
unsaturated fatty acid, we have discussed what were the two types
they
or the
had
three?
they had double bond.
Mm-hmm.
They were glycine.
Sorry, I can't hear you. What you're saying?
What did you ask?
I asked. We have discussed some examples: saturated fatty acid and unsaturated fatty acid.
Yeah,
What are those?
there were
Mm-hmm.
there was I don't remember. See.
Okay. There was, if you remember, there was oleic acid. There was linoleic
Yeah,
acid, right? Those
yeah.
were some examples. Okay. Now, after this, this was a general introduction about phospholipids, and it is mentioned over here that phospholipids are special type of lipids. Each molecule has the unusual property of having one end which is soluble in water. Okay, this is the property, this is the unique property of the phospholipid. Otherwise, we have discussed that lipids are insoluble in water. And why they are insoluble in water? General lipids. I am not
They're
talking about phospholipids. Lipids. Why lipids are insoluble in water?
because of their hydrophilic group.
Because lipids are non-polar in nature. Lipids are non-polar,
Yeah.
while water is polar in nature. So you cannot mix a non-polar thing in a polar thing. You must need two non-polar things to dissolve or two polar things to dissolve. And I told you this concept is known as like dissolve like. Like the like things dissolve other things in themselves. The polar thing will dissolve a polar thing in it. A non-polar thing will dissolve a non-polar thing in it. So that is why lipids cannot cannot be dissolved in water. They cannot be solubilized in water because lipids are non-polar in nature while water is polar in nature. You know, in water there is polarity because of hydroxyl ion and hydrogen ion. That is why water always tend to dissolve polar things in it. Just like salt,
Yeah.
sodium chloride. Salt is, salt is ionic in nature, so it forms ions. So it means it has polarity. That's why it can dissolve in water, but you cannot dissolve oil in water. So this was about the lipid, and then you see each molecule has the unusual property of having one end which is soluble in water, and this end is known as hydrophilic end. The end which tend to solubilize can be solubilized in water is known as hydrophilic, water loving, right? And that is
Yeah.
phosphate group part. And the other region that is in which there are fatty acid chains present, that is hydrophobic region. This one, if you remember, this one is hydrophobic region, the hydrophobic tail, and the head part is hydrophilic. And why head part is hydrophilic? Because of the presence of phosphate group. Okay, because of presence of phosphate group. Clear?
Yeah.
And if I ask you how it is different from a general triglyceride, if you see these two, these are the two images. This is a triglyceride and this is a phospholipid. How these two are different from each other?
It one of the fatty acid is replaced by a phosphate group
In
in the
phospholipid, yes.
phospholipid.
In triglyceride, you have one glycerol and three fatty acid chain. And when you make a phospholipid, its composition is quite similar to the triglyceride. The only difference is that on the third chain, there is no third chain, just like the fat, in the triglyceride. There are just two fatty acid chains, two fatty acid tails, and in the head part, you have a phosphate group. And due to this head part, there is hydrophilicity. Okay, this head part gives the hydrophilicity. And what is hydrophilicity? That it allows water to move. It allows the interaction with water from this side, okay, from the head part. But the tail part, it does not allow interaction with the water because the tail part is having hydrophobicity, okay. And these two nature collectively are known as amphiphilic nature of the phospholipid. You will say this is amphiphilic nature. Clear? So this is
Yeah.
what we are discussing over here, that the phosphate, each molecule has the unusual property of having one end which is soluble in water. This is because one of the three fatty acid molecule is replaced by a phosphate group. Otherwise, we were having three fatty acid chain. Now in phospholipid, you have just two chains, and on the head part, you have a phosphate group attached. That is polar in nature, and that is why it can dissolve in water, right?
Yeah.
Why phosphate group can dissolve in water?
Because it is polar.
Yes, it has
Water
polarity
is
in it.
also polar.
Yes. Then the phosphate group is hydrophilic, water loving, and make the head of the phospholipid molecule hydrophilic. Although the two remaining tails are still hydrophobic. The tails are hydrophobic. Now this is the uh, configuration of the phospholipid. That you have. You see, they have mentioned in this diagram that there is water. So the interaction with water will be from hydrophilic part, and the hydrophobic part will be in the center. Okay. Now there is a concept of mycel formation. What is mycel formation? Whenever you have a phospholipid and you put that phospholipid in water, what will happen? They don't want their tails to touch the water. Why they don't want? Because the tails are hydrophobic in nature. Okay. Because tails
Yeah.
are hydrophobic in nature, so they don't want their tails to interact with water. I'm talking about phospholipids. Whenever you have phospholipid in water, they don't want their tail to touch the water. Why? Because their tails are hydrophobic in nature. So to solve this, what they do? They arrange themselves in a way that only their head only their head part interact with the water. Okay, they arrange themselves in a way that their head part interact with water, but the tail part is intact in the center. Okay, are you getting this? And this is known as
Yeah.
mycel formation. That to solve this, they spontaneously assemble into a circular ball-like shape. Okay, circular ball-like shape known as mycel. It is just simply this is a phospholipid in an arranged manner. Whenever phospholipid is present in water, they don't want their tail to interact with the water, so they arrange themselves in this way, in a circular manner, in a ball-like manner. And in this, if they ask you what is the composition, so you will have this composition that there will be head part outside and the tail part inside. On this is condition only when you are having an aqueous environment outside. Okay, an aqueous environment outside. This is aqueous environment, right? Aqueous.
Yeah.
This will only happen when you have aqueous environment outside. Otherwise, if I ask you, if you have another interaction, like let's say there is in over here inside there is aqueous environment. Okay, inside there is aqueous environment. So how there will be arrangement? Arrangement will be like this. This, these will be the head part will interact with the aqueous environment like this, and now tail part will be outside. Okay,
Yeah.
this is another mycel formation configuration. Mycel formation has two configuration. It depends on the condition in which you are having phospholipid. If the phospholipid is facing water inside like this, the aqueous environment is inside, then the arrangement will be like this, that the head part will be facing the aqueous environment and the tail will be outside. Okay. Otherwise, what I told you is that if outside there is aqueous environment, like this is a condition where there is aqueous environment outside. Okay. Over here, this
Yeah.
hole is aqueous. So in this condition, what will happen? Obviously, it will
Tail part.
arrange like this. Yeah, it will arrange like this, and in the center you will have tails. Okay, because over here there is water. Clear?
Yeah.
Are you getting this?
Yeah.
So there are two types of configurations of the mycel formation. Okay, now, yeah. Okay, are you clear with these two terminologies, the phospholipid and mycel formation?
Yeah.
Okay, and what are the basic? Okay, we must discuss about this. What are the basic functions of the cell membrane?
Mm. It controls the substances that leave and enter the cell.
Hmm. It controls their entering of molecules as well as it has been cell signaling. We will discuss it in detail. And there are many functions. It protects cell membrane also protect the inner content of the cell from the outside environment. From the outside environment. Okay. Okay. Now there is a discussion of the phospholipid bilayer. How phospholipid bilayer is formed and why we are discussing phospholipid bilayer. In our cell membrane, there is phospholipid bilayer formation. The cell membrane of the animal cell is made up of bilayer, the two layers of the phospholipid. This is just one layer, okay? You have two layers in your cell membrane. In this, the hydrophilic head will face outward, the head and the head will be on the outside. You see, this head is on the outside. Head will be on the outside, interacting with the water. This blue color, this blue image shows that there is water outside. While the hydrophobic non-polar fatty acid tail are sandwiched inside. Okay, these two are sandwiched. These tails are sandwiched in between. Are you getting this? That outside you have a phos. This one phosphor. There is phosphate in the head part. So this is known as hydrophilic. This part is hydrophilic. The outside part is hydro.
Hydrophilic.
Yes, hydrophilic because the presence of phosphate group. Okay, and the center. In the center, if you see, if I zoom in, you see there are two layers. This is one layer, okay, and over here this is second layer. So you have two layers of phospholipid. That is why this is known as phospholipid bilayer. Okay, and these two layers are sandwiched in the center. Clear?
Yeah.
Okay, from the fundamental barrier of the biological cell membrane, plasma membrane separating cell forms the cell interior from the exterior environment. The most important function is what? It act as a barrier. Okay, it act as a barrier. The cell membrane act as a barrier for biological molecules. Whether which type of content should be enter inside the cell. Okay, and which should not enter the cell. Clear?
Yeah.
Then the phospholipid bilayer, it can form compartments. Okay, it can form compartments. The bilayer forming the cell surface membrane, establishing the boundary of each cell. Okay, this is very important. Now, what they're saying is that whenever you have different, for example, you have different things in the cell. This is cell, and in this you have maybe there is then you can have and you can have different things over here like I'm just adding different diagrams. And, okay. Now, see, if you see, maybe this is the nucleus. Okay, this is the nucleus. There is this is mitochondria. There is what can be this and what can be this. You can say this is Golgi apparatus. I am just giving arbitrary terminologies, defining them. Then you can have lysosomes, okay, different organelles. What they are saying is that the cell membrane make different compartments. How it make different compartments? Each organelle, every organelle have a cell membrane. Okay, there are exceptions with the ribosomes, right? And there is exception for nucleus and mitochondria as well. That nuclear membrane is double membranous. We have discussed it, right? The nuclear membrane is the mitochondrial membrane is double membranous, right? The
Yeah.
mitochondrial membrane is double membranous. So these membranes, what they do, they help to compartmentalize. They form compartments, different compartments within the cell. Are you getting this? So cell membrane help to form different compartments within the cell. Like
Is that
every
why?
organelle, yeah, because in every organelle, you see every organelle is covered with a cell membrane. So it will have a separate compartment, isn't it? Every organelle will have a separate compartment within the cell. Okay, wait a minute. What is this written? Read this. What they're saying. Phospholipids.
Phospholipid bilayers can form compartments.
Hmm.
The bilayer forming the cell surface membrane, establishing the boundary of each cell.
Okay. So there are compartments they are saying every membrane. You see this membrane. Every membrane is made up of phospholipid bilayer. Okay, and when there are phospholipid bilayer, when you have different organelles, and those organelles are covered with the phospholipid bilayer, so they make separate compartments. And this is how this cell, okay, this cell will also be separate from some other cell. Within the cell, there are compartments as well, and this as a whole, this cell will be separate from some other cell. Why? Because of its phospholipid. Are you getting this?
Mm, can you repeat that?
I'm saying that phospholipid bilayer, it forms outside the cell, right? In the animal cell, when we talk about that, the animal cell is covered with the phospholipid bilayer. So this phospholipid bilayer, it forms a compartment. Okay, it separates the cell from the other cell. Otherwise, if there is no cell membrane, if we remove this, what will happen? All the things of this will be freely available in the blood, isn't it?
Freely
If we,
available?
if you remove if you remove the cell membrane, just like I have done, what will happen? All this content will be available in the bloodstream, isn't it?
Yeah.
So this is what I'm saying, that cell membrane, it separates the inner content of the cell from the other environment, from the other cell, and it forms a separate compartment. And within the cell, you have different compartments as well. Just like there are houses, there are different houses, and in each house there are different rooms. There are many rooms, right? So one house is separate from the other house. That one house have a separate compartment as compared to the other house, and within that house there are different rooms. Similarly, inside our body there are different types of cell, and those cells have phospholipid bilayer in them, and those phospholipid bilayer help to compartmentalize different things. I hope this is clear now. Others,
Yeah,
if you have any confusion, we can repeat this.
I understand.
Okay, this is what. Okay. Yes. Now, internally membrane-bound compartments formed from phospholipid bilayer provide the basic structure of organelles, allowing for specialization of processes within the cell. Now, what they're saying, internally, like within the cell, membrane-bound compartments formed from phospholipid bilayer. Now, they are saying that within the cell, different compartments are there, right? Different organelles are there. So, these compartments are covered with phospholipid layer, bilayer.
They're covered with phospholipid binder.
Every organelle. There are exceptions with there are non-membranous organelles. Can you tell me those organelles which are non-membranous? Like they don't have any membrane.
The the
Hmm.
the rough and
The
rough endoplasmic reticle, the ribosomes.
yes, ribosomes and centrosomes. We will discuss centrosomes and cytoskeleton in detail. Okay.
Yeah,
And there are some which are double membrane bounded. What are those?
and the SCR and RER.
Not the SCR and RER. You must revise that chapter, chapter 1, right? Nucleus
Yeah,
and mitochondria. I just told you, nucleus and mitochondria.
nucleus is just double layered.
Yeah, nucleus have double layer. Double. So there will be total. There are double layer, and each layer is having phospholipid bilayer. Are you getting this? What I'm saying? This is double membranous. Mitochondria as well. It is also having double membrane. So and one layer, each layer is having phospholipid bilayer. Each layer is made up of phospholipid bilayer, like this one. This is one phospholipid bilayer. This is and they are one membrane. This will form one membrane. In nucleus, you have two membranes. Like I can show you. Wait a minute. Double membrane and phospho. mhm. Okay, we can have. There's a complex images. I will show you. Wait for a minute. Just a minute. Okay. Sorry, there was a call. There is. You see, there is nuclear pore and nuclear lamina. I can show you this by. There should be one image. This one. Yeah, this one. If you see this, there is. Where is that? This one. This one is the nucleoplasm, the inside of the nucleus. And this is
I can see.
nucleolus. We have discussed it. And there is this one. This one is nuclear envelope. And over here there are nuclear pores. This envelope, this is the third one. If you see, there are two layers in the third one.
I can't see right now. It's
Sorry,
like frozen. Your screen is frozen.
my screen is. Wait a minute. Oh, okay. Can you see it now?
I can see your screen, but it's like it's on one place. It's written like mitochondrial lysozyme.
Where is that? Isn't it on the main screen where I am showing you the nucleus?
No, it's not.
Okay, then wait. I will copy this image and I will paste it over there. Can you see it now? The nucleus?
No.
No? Now,
No, I can't see.
why is it so? I'm sharing it.
Maybe you should stop sharing and then reshare.
Yeah. Okay, now, is this, can you see this now?
Yeah, I can see it now.
Okay. This is what I was telling you that in nucleus there is double membrane. How there is double membrane? If you see this closely, there is this outer layer, this outer layer, right? And then there is this inner
Yeah.
layer. So this is what I'm saying. And each layer is made up of phospholipid bilayer. Okay, each layer is made up of phospholipid bilayer. So this is what I was telling. You cannot say that these two layers collectively is one phospholipid bilayer. It's not the case. These two separate there are two layers and each layer is made up of phospholipid bilayer, just like this. Okay, just like this one. Okay, this makes one layer. This is three-dimensional arrangement. This is not this. You can say this is looking like a sheet-like structure. So this will
Yeah.
form one membrane, and in nucleus and mitochondria you have two membranes. So those two membranes they collectively form the outer membrane of the nucleus and outer membrane of the mitochondria. Now this is what I was telling you that internally membrane-bound compartments formed from the phospholipid bilayer provide the basic structure of organelles allowing for specialization of processes within the cell. Okay, so the cell membrane will help them to perform their special functions. Like we have discussed the function of the endoplasmic reticulum, the ribosomes. What are the function of ribosomes?
Protein synthesis.
Yeah. So they different organelles have different functions, and that is why they are specialized. Their membranes are specialized. An example of membrane-bound organelles is lysosome, found in animal cell, each one of which contain many hydrolytic enzymes. And what is the function of hydrolytic enzyme? I told you. What are those?
Um, it was the
Hmm.
toxic. It's
Yeah,
toxic
hydrolytic.
and old guys.
Yeah, hydrolytic. Lytic means lysis, and hydro
Yeah.
means in the presence of water, aqueous. It is from aqueous. So in the presence of water, like in the presence of the aqueous content of the cell, these enzymes they break all the toxic substances within the cell. Okay, so the lysosomes have hydrolytic enzymes and they break down different kind of biomolecules as well. And why there is need to break down biomolecules? Hmm? Why there is need to break down biomolecules? Like for example, you have a carbohydrate and you get glucose from the carbohydrate. So why it is needed to break down the glucose?
Can you repeat that? I couldn't hear you.
I'm saying that why the it is mentioned over here that these hydrolytic enzymes they break down many different kind of biomolecules. My question is that why we need to break down those biomolecules? Why there is need to break down these biomolecules?
Um, because to use the energy they have,
To use the
energy they contain, to break them down
yes,
to a usable form.
because by breaking them down you will get energy like by breaking the lipids and by breaking the like, for example, when we talk about the carbohydrates, in carbohydrates, if we talk about the simple carbohydrate, the monosaccharides, they will give you glucose. And your cell cannot absorb glucose directly. Like your cell cannot get energy. Absorption can be done. The blood can absorb glucose, but it cannot get energy from the glucose. So the glucose has to be converted into ATP. The ATP is the energy currency of the cell. That is why the glucose will be breakdown. Okay. So this will be done by different hydrolytic enzyme will help in breakdown of these molecules as well, and also to break down the toxic substances as well. So this is the specialized function of the lysosome. This is what they are mentioning over here, that the cell membrane, the phospholipid bilayer, this help to uh, compartmentalize, to differentiate. You can say you have these organelles are different from each other. You cannot say that these are similar to each other. They all are performing different functions. So that is why we are saying within the cell there are different
Yeah.
compartments and there are different organelles. Those different organelles are performing different functions. Their specific functions. Okay, are you clear with this?
Okay.